Thank you for joining us today for our GROW webinar. It is my pleasure to introduce Dr. Kauê de Sousa. He holds a PhD in Applied Ecology and Biotechnology from Inland University in Norway. He joined the Alliance of Bioversity International and CIAT in 2016 as a research assistant at their Costa Rica office. With over a decade of experience in international agricultural research, Kawa has made significant contributions to various areas aimed at transforming food systems. His work has focused on farm innovation and experimentation, data integration, software development, and the agri-food interface. Kawa has played a key role in mainstreaming the Tricot approach, a digitally supported method for on-farm testing in Africa, which has benefited over 30 teams across 15 African countries. He is also committed to capacity development, mentoring students, and training professionals in decentralized agricultural and data analysis. His presentation today explores how the Tricot approach and ClimMob software are enabling a shift in the way germplasm is evaluated, moving from a centralized researcher-driven trial to farmer-led decentralized testing. The title of his talk today is From Genebanks to Farms: How Citizen Science is Transforming Crop Variety Evaluation Through the Tricot Approach. Welcome, Kauê.
Thank you everyone. So good morning, good evening, good afternoon, whatever you are. So for me, it is a pleasure to be part of this webinar to share a bit of our experience, over the last, let's say, 10 to 12 years, developing and validating and also scaling the Tricot approach. Here in this presentation, I will talk a bit of like just a small scratch of what we have been doing, and then also I try to frame the presentation on the framework of gene banks to farms. And of course, I like to always recognize that this is a citizen science approach, and the picture that I chose in the portrait of this presentation is not taken as like by accident. This is a amaranth trial in Tanzania, which was 100% led by this farmer in the close to Arusha. So this was part of one of the projects that has been supported by Crop Trust in Norway, BOLD project. And the idea is that the farmers are at the center of the trial. They are the ones experimenting. They are the ones telling to the scientists how this works in their own conditions, and also contributing to a large scale on-farm trial network. So in this presentation, what we will have is we will talk about the challenges that drives our research, the Tricot approach, and also 5 state case studies on and lessons that we learned. Also, it's important to say that some of these slides were borrowed from my friend, my colleague, Janet Steinck and Jacob van Etten, who are the forerunners of this approach. Back in 2013. So this is some of the lessons that we have been doing the last 10 years. So of course, when we do Tricot, we don't do it by any. It's not it has a purpose, for doing that, and we want to address a specific group of challenges. The first challenge that we have is loss in agrobiodiversity. This is one of the core things. And actually, one of the things that we started. One of the reasons why we started Tricot. Now it's important that recognize that we have many crops that people can grow, but of course we have very few that researchers focus the time and investment on some very few number of crops, and of course also very few number of varieties that are grown by farmers and or promoted to farmers. Another issue that we want to address is climate change. Climate change can have many sides, many effects. For example, in the north, it's very likely that farmers will experience good conditions to grow crops, but also increase of pests, like new insects that can come. Probably in the south or in the other regions, farmers will experiment more dry seasons, or maybe more diseases, caused by rainfall or other issues. So climate change is not a single issue, and it's not easy to address as a single solution. And then we need to also cope with that. So this is one of the challenges that we try to address with our methods. Another issue, which is very common, but very few people talk about that, is about issues in data capturing. Because in my perspective, I work in the group of digital inclusion, collaborate with many other partners in across the alliance and also across the CG in what we call data-driven agriculture. And the idea of that is because we want to approach this issue, of data capturing. For example, most of the trials that people do, they are designed to fulfill a single objective. The people designing the trial, they want to fulfill one goal for that they want for a specific project or intervention. And sometimes this happens that most of them they will develop or implement trials that are geographically dispersed and that has very limited opportunity to aggregate the data or repurpose the data, because everybody implements trials or implement experiments under their own ways, and then it's very difficult to put them in a context when you need to, for example, do a regional analysis. And then there is also very little in long-term focus on the opportunities that a well-structured data can provide to scientists. So in this presentation, of course, the objective is to show with a case study of agrobiodiversity and use of crops. But the goal here is also to say that when you have a well-structured data approach or for collecting data, on what you want, you can have more insights and you can provide more recommendations to the end users. Now, in that case, the end user here that I'm talking is the farmers and consumers of agricultural products. So that's the overall goal. And I think that people take very little attention to that specific detail. So we saw, and there's the evidence also demonstrate to us that the increased agrobiodiversity is part of the solution of at least for objectives 1 and 2, the challenges 1 and 2, because different varieties, they have a different climate adaptation, so they can adapt. Weather extremes will not affect all varieties equally, so this is one thing that we also know. And also growing multiple varieties together, in a portfolio also can increase climate resilience. But how can we support farmers to find varieties that match the conditions in their farm? So in Norway, we have the say that there is not such a thing as a bad climate. Just It's not such a thing as a bad climate. It's just bad clothes, no. So if you take this as in the context of varieties, there's not such a thing as a bad climate, just inappropriate varieties. So this is we need to use the proper varieties, for the specific context that we want. Of course, also, but to get this information, we need to have participation, we need to have feedback, and we need to do trials to know how these varieties perform in different conditions. And one of the issues is that most of the trials, they are implemented in centralized locations, and then it's very difficult to extrapolate the results of these findings or the findings of these trials towards a more context-specific conditions, for example, at the smallholder level, for example. Another thing is also farmers, when they participate, often what the evidence shows to us is that most of the farmers, they do inconsistent observations because they are not participating all the steps when people do participatory evaluation with the farmers, mostly because of logistical conditions. Not the farmer maybe is not interested in one of the crop stages of the crop. So this led to farmers not participating and not engaging in the trial as we would expect. So, and also we know that farmers, this the consequence of that is that farmers will have lack of ease access to variety, to diversity. So for example, from the seed system side, one of the issues here is that there are very few incentives to produce only few varieties at large scale. And also the seed system, it also lacks information on the variety demand. So they don't know which variety the farmers need or want. And also farmers, from their side, they also lack information from the available varieties, the varieties that are out there. Sometimes the farmers, they rely in a very specific number of varieties because they already know. And for them, of course, is a very risk approach to take out the varieties that they are planting and change it to new varieties because they don't know what will be the performance of these varieties under their conditions. And one thing that we know is that for farmers, no, and for many other groups of human beings, seeing is believing. So then farmers, of course they will get more trust on the varieties by learning, by doing this, and then getting to know how this performs in their conditions. Possible solution for that is that farmer managed trials can, empowered by citizen science, can enable this and also can give information to seed systems or gene banks or whoever needs information about the seeds demand. And also farmers can get access to new materials and also they can test it and see if they want or not such materials. With that reasoning of thinking about that, we developed the Tricot approach, which is short for Triadic Comparison of Technologies, which is a citizen science approach that can help bring in diversity farmers. And during this presentation, I will show you how citizen science, how Tricot can help on that. So this is not it's not the only solution. What I want to say is that Tricot can be the enabler for that, because of course there are many institutional barriers and also other types of barriers that needs to be overcome so then people can get access to this. But Tricot can be the enabler, can be the one creating evidence on it. So then we can provide more, let's say, institutional insights for that. So Tricot is not something like, let's say, is not something that is out of this world. I think meaning like this something that it's something that is based on the best of existing approaches. No, it's that's the beauty of the approach, because it's a novelty is by bringing together different approaches that can be by being connected, they can address specific needs of each group in this chain, in this, like I didn't mention anything about the chain or seed chain or value chain of crops, but this is basically many people, stakeholders, farmers, seed systems, gene banks. So there are many actors involved in that. And then by bringing together all these approaches, we can address these needs, and then we can also inform each of the actors. One of the things that is key in Tricot is the citizen science. No, farmers they they are the ones that generating this data, contributing with data, and citizen science is something that is not new in its sense by being employed by Tricot. Citizen science has been being employed in many other topics, no, and the most famous one is in bird ecology, when you have people that are, let's say, enthusiasts about watching birds in the nature. They take pictures of the birds, they identify the birds, and they send it to, let's say, a common database that's managed by one of the most famous one is in Cornell University. So they manage this database. The people send the data there, and these people, the researchers doing the assessments on bird migration, climate change effects, so all these things, they get access to this data, and then they can see, for example, how climate change can affect bird migration. And if you see, most of the data that are used in ecology, bird ecology, is not generated by, let's say, researchers with a PhD, but basically is generated by citizen scientists, people that are not formally trained in that topic, but they know about that because they are engaged in this. No, so basically this is the same thing with farmers. They are engaged in farming, they know a lot, and they can contribute a lot with that. Another thing that is we employ a lot in Tricot is the concept of on-farm testing, or basically testing under local conditions. So this is to enable, to capture the performance of under the conditions that the species are growing. So this is very common in North, like in US or in Norway, where farmers they collect the milking records because they need to know that to know how much each of the cows are producing. So this information is collected for free and for free for the researchers, and the farmers share this information with the researchers. So then the researchers put this into the genomic selection models to see which are the best bulls, for example, for bull breeding, for example. So of course, this is just one species, not two species of bulls. So it's much it's less complicated than having many crops and all these polyploids and diploids types of crops. But the idea is the same. We also employ concepts of digital solution. I'm not talking about flying drones and all these very complex ways of doing digital agriculture, but I'm talking about things that are very easy and handy for everybody to do, like having a very well-structured trial design, a software that can receive the data and can also give reports to you, and also using novel digital tools and analysis approaches. So this is basically all this idea on the digital agricultural side. And of course, participatory research, which is basically putting farmers at the center of the evaluation. And this is the photo that you see here on the participatory approach are farmers in Ethiopia assessing the performance of durum wheat that were obtained from the gene bank in Ethiopia. So basically the Tricot approach works in that way. We have designed one platform called ClimMob, which is basically the brain of the approach. So everything should be designed there. So I always say that if you don't design your Tricot trial on ClimMob, you can call your trial whatever you want, but you cannot call it Tricot, because basically ClimMob is the one that will give to you the randomization, will give to you the structure to collect the data, and will give to you all the support for that. So you can design whatever trial you want, but if you do it outside ClimMob, you are not doing a Tricot trial, and I will tell you why. The first reason is that when you select, let's say here, just for the sake of fitting in this screen, we have here five varieties, no? And then by colors. And then what ClimMob will do for you, will pick out of these five varieties, will select three, no? So each farmer, no, each participant will get three varieties out of the five that we have here. So this is in statistics, we call this design an incomplete block of three. So each farmer will evaluate three, and also ClimMob makes sure that I have this in my next slide, but ClimMob will make sure that all the varieties they are tested in a balanced way. So every variety will have the same proportion of testing, and they are being tested equally, and also in a very specific order. So then even if you lose some packages, or if some farmer drop the experiment, you don't break the network of the experiment. So you can still keep the experiment going on, and you don't lose your data. So that's why I'm saying that this needs to be done on ClimMob. So then you have the package. Let's say you selected your five varieties. You went to ClimMob, then you select how many farmers are going to participate. ClimMob will generate for you one randomization for each of the packages. Each package here is a farmer. So then you can have four, but you can have 400, 4,000. There is no limit. So then you have here, and then you make the package with the seeds, no? And then another thing is that they are labeled ABC. Basically, is a blind trial, because we want the farmers, in most of the cases, depending if it's a seed system trial, mostly, we don't want the farmers to know the name of the varieties that they are assessing in the beginning of the trial, because we want to avoid bias in the evaluation. So the farmers will know the name of the variety by the end of the trial, when they get the results of what they did, and also they compare with what the other farmers did as well. So they will know what they evaluated, but in the beginning, the idea is that they keep it blind. And in most of the cases, people ask me, but this is what if the farmer wants to know? And basically, this is one of the motivation factors for the farmer to stay in the trial, because they want to know the name of the variety they are testing. So they are like all the time curious to know, no? And then they talk with the neighbors to compare varieties. So this is basically one of the social dynamics that also Tricot enables. So then the farmers, they are registered. Now we collect just data that will help us in understanding, knowing who the name of the farmer, and getting contact with the farmer in the next trials, in the next stage of the trial. And then the farmers will plant it side by side, A, B, C. Each crop has a specific protocol for the plot size and also the number of seeds that each farmer receive. And most of the crops they have, but for some crops, we still engage with the stakeholders to develop the protocols to design these trials. And then they plant it. And then at the end, and then when we have the traits that we want to assess, the farmers will assess the traits. Most of the cases, they will do a ranking. So for example, we will ask the farmer which variety, for example, let's say yield or marketability. We ask the farmer which variety has the higher yield and which variety has the lower yield. So that's why Tricot is 3 and not 4 and not 5 and not 2, because 3 is the optimal number that allows you to make two very simple specific questions, or two very simple specific questions that will enable them to focus on the edges of the ranking. No, so we are not forcing the farmer or making the farmer to make a ranking. For example, if I ask you to make a ranking of 5 things, the best 5 movies that you have seen in last year, for example. So it's very difficult for you in certain points to put these rankings in an order, because maybe one movie was very good, and then for you it's difficult to place them in a ranking. So this requires lots of cognitive attention and lots of cognitive effort. So to enable people to participate on that without making lots of efforts to make the ranking, we do the tricot, which is basically a best worst scaling. You just focus on the best and you focus on the worst. And then we know by default which one is in the middle. So this is a very simple solution, and then everybody can do it. And this is one of the nice parts as well. But we not only collect rankings, so now people can also collect yield data in metric scale. So ranking is not only the what we do with Tricot, but is one of the easiest way of collecting data, and also straightforward way of collecting, because you can easily aggregate and you don't need to stay many time cleaning and also debugging the data in case of any specific things that can happen. So that's why the focus is on rankings, but it's also possible to collect metric data. Later on, the data is aggregated using as well the ODK collect, which is the tool that we have. It's not developed by us, but it's developed by a third-party group. But everybody can use it, so we also take advantage of the open software for that. So we use ODK, and then the data is later on sent back to ClimMob for analysis. So ClimMob basically do uh, specific analysis for you, showing the performance of the trials and also the varieties that you test. And then the next step, which is not here in this slide, is giving feedback to the farmers. So going a bit back on the incomplete block design, this is how the incomplete block design works on ClimMob, and that's what I'm saying, that it is very important that you design your trial there, because you have this scheme, where you have the varieties being tested randomly, but also in a very very well structured. So for example, if I see here the first 10 packages, each row here is a package, and then you also see them reflected here by each farmer. And then you also see that there is a check variety, or sometimes people call control, or whatever they decide to call, but generally is in the context of a variety. For example, this would be a variety, a local variety, or a variety that the farmers are most popular variety in the seed system. So one of the things that we say is that this variety is also embedded in the trial. It's not like one variety being tested in each block, no, like all farmers are testing the check. We don't want to do that because by the end, if everybody tests the check, you are creating a bias towards the check, and then you are getting to know more about the performance of the check variety rather than the performance of the variety that you want to introduce or to want to expose the farmers. So, but in some places, what people do, and this is very common in maize, for example, is where the farmers in maize and in cassava as well, is that the farmers, they put here a fourth plot, which is just an observation plot. It's not part of the design, it's just a comparison plot where the farmers will use the fourth plot as a benchmark to compare against the three that they are testing. But this is not part of the design. But at the end of the trial, what we do is that we ask the farmers to compare. So then we ask them, what was the best one, letter A or the local? Which one is the best one, letter B or the local? Which one is the best one, letter C or the local? So then they do like this pairwise comparison, and then we know from their perspective whether the local variety is better or not than the Tricot trial. But this is not embedded in the randomization because we want to avoid this bias. Tricot in practice works in that way. First of all, you choose the variety that you want to test. You make sure that they are multiplied. So generally this work is done, I don't know, ideally 6 months before the trial being implemented. So before the beginning of the crop season, ideally 6 months before, or even 1 year, depending on the crop. So then you choose the lines, so the varieties, the genotypes that you want to test, and then you multiply them, and then you make sure that you are getting enough seeds for that. And then later you randomize, you know how many farmers you will be working with. And then you randomize this, you prepare the packages, and then you have here, this is a package from being trial in Nicaragua. So then you put them in these bags, A, B, C. And another thing that is nice as well is if you can distribute these small flyers with information for the farmers, no information here on what is the goal of the trial, what we want to achieve. One thing that we also put sometimes here, depending on who is implementing the trial, is the consent form. Sometimes the consent form is already embedded here in the flyer, or they do the consent form independently from that. So where the farmer will tell the scientists whether or not they want to contribute to this trial. And then here is the evaluation card where the farmers will put the information that they will be collecting. So this is one way of collecting data. The other way of collecting data is that the extension officer goes directly to the farmer or call the farmer and register the data using ODK. So these are the two ways. I also have another example. And here is a picture from Boulder, no, the Boulder project. This is Jute Malo in Benin. And then here you see the variety names, no? But here is because this is what the people in WorldVeg were doing to get like a validation or to verify the variety if the varieties were placed in the correct place. And this is the QR code that I mentioned, no? The code that is the ID of the package that is generated by each of the incomplete blocks. Generally, the trial should be done side by side. So this is a bean trial. In that case, for beans is 6 meters long with 4 rows. No, so 4 rows of 6 meters. They should be planted on the same day. Uh, Generally, we don't have big plots because we want to allow comparison, and also we don't want to marginalize farmers with a small that has a small land available. So the idea is that we have small plots that are easy to be managed. And also, this should not be an extra work for the farmer. No, the farmer should see this as an experiment. It's not like doing a work for like it should be something that they are happy to do and proud of doing. And this is basically the case in all the trials that we've been implementing. Ideally, the farmers, they will do the same management that they do to the main plot. The main plot is where the plot that they grow their local variety or they do their local trial. And this is one example. No, it's different from the one that I mentioned before, but this is one example of jute mallow in Benin. So here you have the each plot, A, B, and C here, and then the farmers are now doing the management, removing weed, and then also they put a fence to avoid the animals to come in, but and also they have this is separated. No, they have a space between them. But this is basically how one trial is done for other type of crop. So as I say, the type of trial, the way that the trial is done, and the size and all these details are very crop specific. So this is informed in the standard operating procedures for each of the crops. And we have that for most of the, let's say, the most popular crops. And now we are moving towards having SOPs for the underutilized crops or opportunity crops under the framework of the Boulder project. Something that I forgot to mention is that in this here, no, it's basically gene bank accessions from WordVeg. So these are the names of the accessions here. So I'm talking about Tricot, but you already see varieties coming from the gene banks to the farmer field, no? So basically, these are gene bank accessions being tested by farmers, and these are basically informing the breeders and also the gene bank managers how a variety performs on the local conditions and if they should advance it or promote it in a seed system scale or even consider a breeding program for that. So then farmers, they do very simple observations. No, it's just replying which one is the best and which one is the worst. You can do it in the card. And also you can have the extension officer assessing this with the farmer, so they don't influence the farmer in the choice. They only are there to visit the farm and then ask the farm what the farmer thinks about each of the varieties for very specific traits. Also, this is under the framework of Boulder. This is Anna Mlak. She's a researcher from WorldVeg, so she was doing the work in that day, checking with the farmer about the performance of these amaranths. The farmer will, so for example, for some of the crops, this is beans. We have here the farmer evaluating which is the variety that has the best leaf development and which one has the best winding. So then the farmer will tell which one is the best and which one is the worst. As I already said before, we know by default which one is in the middle because they focus on the best and worst scaling. And then we can place them in a ranking. So this is something that is very straightforward to be done here. And of course, the traits that are assessed here, in most of the cases, they come from interactive approaches, like more participatory approaches as well, working with the farmers and understanding which type of traits are important for them. Also, Tricot is nice because Tricot can do backwards prioritization of traits. Because if you test, I don't know, in 2 cycles, if you test, I don't know, lots of traits, you can later on do a backward analysis and see which are these traits are driving the overall preference of the farmer. And then you can start reducing traits in the next interaction with the farmers just by knowing which are the traits that are driving their choice at the end of the trial. And something that is important is that we have different traits. We have different traits being assessed, but a trait that is mandatory in the approach that is in all the trials is the overall performance, because this is basically the main trait. We ask them overall after, I don't know, 3 months assessing, evaluating this trial. Every day you are there. So overall, which one do you like most and which one do you like the least? So this is the overall preference, and this is the trait that helps us in assessing the drivers of a farmer's preference on the traits. If you see here, for example, it's very classic. This one, it's nice to see that each variety are phenotypically distinguishable. And also you see that they have different sizes and different shapes. And this is something also that also make the farmer assess and prioritize different genotypes. And again, these are 3 genotypes that came from the gene bank at WorldVeg in Benin. The data collection can be done today using at least two approaches. ODK apps, so basically what Anna was doing, in this picture here. She was using paper, but also it's using the cell phone. So you can do ODK, or you collect the data on paper, and then later on you submit to the tool using what we call Enketo, which is basically a feature to collect the data and submit it in the browser. The data workflow works in that moment. So now I explain how the tricoter works. I explain some of the principles and some of the practical things that happens. And now I'm telling you now the full picture. Now, not only from the let's say this field research, but also looking at the let's say the more from above when you see everything that happens even in the back end in the software. So the first thing, of course, is that we have the farmers. Now we have different farmers generating data. So they will contribute with their very specific tasks, and to this big task, and then they will inform the trial manager with the performance of the plot, or the crops, using rankings. So then ranking is very easy to aggregate, so we can aggregate that. So then we know now we have a full data set, with the performance of all the varieties here. So this is basically the ranking. So with this ranking already, we can analyze and we can do lots of analysis, and we can do lots of insights out of this, because we can know at least which variety is the best overall in the project. But what we do as well is that we integrate this data here with other data sets that come outside of the trial, and sometimes they use parts of the information in the trial to inform better. So one of the information that can come is the GPS, using the GPS, the location of the plot and the planting date, is that we can easily collect climate data to see the performance of the varieties under different climate conditions. Another thing that we can use is management practices, socioeconomic characteristics, and also laboratory data, like DNA or even instrumental data. Today in this presentation, I will show to you 5 case studies where we use this type of data, knowing in each of the cases. Most of the cases we did focus only in climate, other only in management and social, and other one only in biophysical data. But now we are trying to put them all together in a framework where we can have a better overview of everything. So then the data is linked to that, is analyzed. We analyze the data here, and then we produce the reports automatically. So this process is automatic. And this reports are produced automatic. So one report goes to the farmer. So the farmer will receive the feedback after the end of the trial. And also the trial manager will receive a report as well with more insights. And then most of the cases, they come to us and request for additional data additional analysis to test some hypotheses. And then what we do is that we do it locally, and test, and we validate with them what are the main questions. And then if the data and the code can be generalized, we send it back to the analysis here, so then everybody can benefit. So basically, this is not only participatory research here, but also participatory research here, where we have this interaction with the scientists in getting this thing moving and advancing more with new insights. At the end, of course, we have the feedback to farmers, which is an integral part of Tricot, and because the farmers, they are curious to know what they test, and then they want to know, and this is very important that people budget for that when they are doing the Tricot planning. And then this is basically the feedback can be done by village. Generally, people implement the trials in specific villages, like if they are doing a country level, they generally choose a certain amount of communities or villages, and they have, I don't know, 10 to 20 farmers per village, and then they do this by these groups, and then they invite them, they share experiences, and they see the results. Under the framework of Boulder, we are testing this new feedback to farmers, which we think should be more, let's say, user friendly for them to understand. So this is something that is under development right now, but we already have a feedback report ready for them, and then we are trying to improve it. It's very important to say that Tricot is not something that is done like only Bioversity or CG thing. It's basically a multi-institutional partnership. So we have people contributing over the last 12 years to that. So it's many partners in Africa, in Latin America, of course, receiving support from many donors, no, like many donors, most of them here in this side here of the the slide here. And everybody here are people that are implementing and also engaging and contributing to the approach. And something that I'm really proud of is that last year, no, we came with this publication with 84 co-authors. Basically everybody that contributed to Tricot in some way, people that did feedback, that contributed with new ideas to improve the approach or to engage with the farmers. And then this is basically a paper that I really recommend as a, let's say, first paper to read, why we took some decisions on doing Tricot and why we did why we do what we do in the approach. Because basically this is reading all the experience over the last 12 years. Now we went to the, let's say, to the theory of Tricot and also some experience that some tips that I have on the experience that I gained from using implementing the approach. But now we are going to do the case studies that I promised. So the first one is a case study that of a very long-term project that we had in Central America, working with common beans. Common beans is a staple crop in Latin America in general, and in Central America, I think it's even more. And then we did this with genebanks and also breeding sessions, from breeding lines from the countries that we did. So basically, we see here every point or every color here is one trial that was implemented in these countries. So we have El Salvador, Honduras, Nicaragua, and Costa Rica. And then they tested 17 genotypes over 12 seasons. And in total, there were 305,000 farmers. Oh, yeah, 3,500 farmers testing these varieties over these 12 seasons. So generally about 250 farmers in each season. And also the farmers were testing basically how these varieties performed, and also comparing against release varieties. Now the idea of this project here was a seed system trial that we wanted to see how can we could increase diversity on farm with varieties that were resistant to drought in the region, because basically all this area, from here, if you see my mouse here moving, so all this here towards the Pacific are dry area now, they are really badly affected in some years by drought. So the idea here was to increase diversity and make them more resilient to drought by having dry resistant varieties. So what we got here is that we, following one of the approaches for data analysis, is that we got the rankings that the farmers generated with climate data now, because we have the GPS and we have the planting date, so we can easily extract the information from public available the climate data. One of the main data sets that we use is called the CHIRPS, and one of the other data sets is called ERA5. So in this case, they use the ERA5 to get the climate data and create climate indices for the varieties. And then what we saw here is that basically this climate index is the wet spell duration index, is the number of days with a consecutive rain during the crop growing. So this is one of the things that affect the crop performance. And what we see here is two climate scenarios. One where you have a very dry area, and then when you have a more, let's say, more humid climate, where you have more rain. So basically the performance changes. No? So here you have the performance. So as far it goes to the right is the best, the performance. And here are the variety names. So then you see here clearly, that we have here three varieties that are do really well during wet conditions. And here you see a group of varieties that do really well under dry conditions. But of course, here you create only two scenarios. And also, since we have the GPS and we have the planting dates, we can extrapolate this data to create recommendation maps. So basically what you see here is the recommendation map for one specific season in Central America with the best three varieties in each of the areas, now in each of these small grids here. What is nice here is that we are not recommending a single variety, we're recommending a portfolio variety. And something that we are now under development is to get to know the proportion of each of these varieties in the portfolio. So for example, maybe in a more dry season, you were not going to plant all the three equally. Probably you want to have a higher volume of this variety, and maybe 50% of this, 25% of this, and 25% of this. And then in another season, you can change the portfolio and have more seeds from one variety to increase your chances to have to don't lose or to reduce your risk of losing yield due to climate conditions. So this is one of the, let's say, the key insights that we can get from these variety portfolios that can be enabled by Tricot. Now, so this is basically something that can inform the seed systems on how to prioritize varieties for multiplying in the next years, and also inform the farmers which variety are very likely or more likely to be used and also be well adapted to their conditions in each of these countries. And this is nice because it even in areas that we didn't test, we could also extrapolate. And this map here shows where we cannot predict. So basically, this is areas that people don't grow beans. This is highland here in Costa Rica, and this is basically mangroves in the coast here. So of course, the applicability of these recommendations are basically zero. Another case, which is basically one of the first cases of Tricot, is when we did durum wheat trials in Ethiopia. So in this case, the idea of this project is that they wanted to restore the breeding programs, and also restore diversity of crops, crop use in Ethiopia, because most of the farmers, they were using a very specific group of Mediterranean varieties that were not really well adapted to the climate conditions of the country. And the idea was to bring back diversity. So this was under the Seed for Needs initiative. And then we did it with durum wheat, Ethiopia, and 41 genotype accessions from the Ethiopian gene bank. So all of them were genotypes plus 3 improved varieties. And then this was done in 3 seasons with 100,000 farmers, basically 300 farmers per season in 3 regions of Ethiopia, no? And then this map here shows to you where these 41 genotypes came from. So you see that we have a very high, at least a very high geographic diversity of gene bank accessions, no, or passport data on gene bank accessions. So we have here, but most of them, of course, coming from the highlands in the country. So this is already something nice because you see at least the latitudinal diversity here. And what we learned from this trial was also joining with climate data, no, is that we learned that we could we could also find climate scenarios where the varieties had different performance. So here you have a minimum night temperature above 11 degrees or below 11 degrees Celsius. And also you have the maximum night temperature, which is below 12 degrees or above 12 degrees. So basically you have here let's say cold, warm nights. You have here warm nights, and also here you have cold nights but with warm days, and also cold nights with cold days, no? So this is basically cold conditions, and this is warm conditions, and then this is even warmer conditions because here it's like you have a very high gradient where the temperature is low in the night and also high in the day. Uh, so then, so then you can see here. This is the blobs now from the gene bank sessions, where people collect the SNPs and then we put these. It's not blobs, it's SNPs. So then we plot it and then to see the genotypic diversity. And then what we see here is that the three lines, no, the three sessions that I mentioned about being let's say, the current recommended or the commercial varieties in the country, they are really far away from the genotypic diversity of the other ones from the gene banks. So basically, all these are gene bank sessions. And then out of the gene bank sessions, we could identify two varieties, no, or three varieties in each of the groups here. We split in, let's say, in cold tolerant and warm tolerant varieties. And then we could find three varieties out of in these two groups that could be really recommended for the farmers under with local conditions or local adaptation. So this is something that is nice. No, and if I recall well, one of these varieties was actually released. You know, they take it out from the gene bank, they assess it, they did some purifying of it, and then they managed to release it as a variety for the farmers. So this is this is the second case study of where we are getting varieties from the gene banks, sending out to the farmers in a large scale trial over 3 seasons, and also merging with climate data to understand why this variety performs in a certain way. And then you can inform the seed systems, all the stakeholders involved, and also the farmers on the best variety for their local conditions. Another example also comes from Ethiopia, is about diversity and seed systems. This is not basically an example for, let's say, under the context of this variety, under the context of this presentation, but I like to show this here because, first of all, it shows two things. No, it's more about gender specific issues where you have different genders playing different roles in how they choose varieties. So this is something that we need to recognize as well. So here we have the share or the proportion of varieties, the number of times that one group of farmers, the men or the women, chose the top 25 varieties in the sets that they were testing. So we see here, now this is the agreement. So ideally, one agreement should be higher than 75% to be, let's say, so then we could say, no, men and women, they agree in the varieties that are the top varieties. And this is not the case. So if you see here, for example, in most of the cases, the agreement is below 50%. Now, this means that when a man choose a top variety, the women will choose a totally different variety out of this one. And of course, there are reasons for that. We couldn't analyze that in that specific project because the data could not be used for that. But what we could show here is another thing, in which brings to the context of this presentation about diversity, is that the farmers, they are not people always say, no, the farmers, they prefer to still grow local varieties and they don't like new varieties, which is not specifically true. The farmers, they prefer a portfolio of varieties. Now, basically, this is what we found here. They prefer a portfolio of varieties. Say it's very what we saw here, very common, is that the farmers, they like to keep the varieties that they still grow, and also they want to include the new varieties that were introduced to them in the portfolio. So this is something which is really nice and also important from this project. Another one is that men and women, of course, they have different trait preferences, and farmers they will favor a diversity number of traits. So basically, they are focusing not only in yield but also in other traits like marketability and taste, for example. And this one is analysis that did, let's say, a dichotomous analysis, like a dichotomous approach, only doing men and women. And I'm going to give to you the fourth example, which is also, let's say, more dichotomous. On you have two groups of two different socioeconomic groups uh, showing the performance, showing the preference towards a specific crop. And this is basically from cassava in Nigeria, 10 genotypes and 1,000 participants. This is not on-farm testing, but this is also a case that I decided to take to this presentation to show to you that Tricot can also be used to do consumer testing. So basically what happened here is that the consumers, they evaluated 3 out of 10 samples of Gari Eba. So if you don't know what is Gari Eba, Gari is basically a cassava subproduct, is a flour from cassava. It's very common in West Africa, and then it's a staple food in West Africa. And then people transform this into eba, no, which is basically kind of a small dough of Gari, and it's very important for them. So they did this consumer preference there in Nigeria with these 1,000 participants. And also we did linking, farmers' socioeconomic data with breeding data. And what it basically, this is, let's say, a further step to this, the dichotomous thing. This is a further step to this, because here what we did is that we, first of all, we try to see if the ethnicity, we tested in different types of socioeconomic characteristics, but the one that showed to us the significant influence on the ranking was the ethnic group. So what happened here is that we found that two different ethnic groups have a preference towards different types of EBA. So for example, if you are from the Idoma, Igebe, Yoruba communities, basically you reject these two varieties, but you will prefer all of these varieties here. These 8 varieties are among the ones that you prefer. But if you are from Hausa, Igbo, Igbira, or Tiv, basically the variety, the genotype is not variety. The genotype that you prefer is this one, only this one. Okay, so now we know that ethnic group has a significant influence on how people choose varieties. But what can we do with that? I don't think there is much to do here because we don't know why. And then what we did here is that we pick the socioeconomic data, the ranking data, and the breeding data, which is the biophysical data that you collect in the laboratory, let's say color, stretchability, moldability, all these things that you collect in a controlled environment for the samples that are being tested. We linked that to the ranking to understand why these groups are taking these decisions. So then this is what we have here, and if you see, all these things are published, so you can easily access all these using these DOIs here. So what we found is that for the first group, solubility is something that is really important. And what I even validated that with some Nigerian colleagues. And what they say is that basically people from this group here, the house, all these groups, they prefer Gari with a higher, let's say, it's more, they prefer sour taste. So the solubility should be lower, should be very low, should take time to soak. So then it starts to get a bit more sour. So that's why this variety was the one preferred by them, because it was the one with more sourness. They could taste it. The other group in the other way, they value more the color of the Gari, because these groups, they tend to put palm oil to the EBA, And then if the color is yellow, so the color will not change so much. So they prefer agave, which is more, let's say, white, because then by adding palm oil, they can color it. So this is one of the findings. So here we managed to find the how, no, how people choose. And here we managed to find the why people do this choice. So this is again another data approach or analysis approach, again using this dichotomous analysis, where you have one group or another. So what we are moving on, and this is something that we did with Amaranth, which is the case study number 5, is basically going away of that and basically classifying the group not in dichotomous things, but creating a specific segments or specific characteristics that they could be more familiar with. So the idea here is that we could create a kind of a persona-based approach where even if you don't share 100% of the similarities with that group, but maybe this group is the group that you are more familiar with, the group that you feel more identified with. And this group can embrace everything. You can put climate information, you can put gender, you can put market segments. But in that case of this paper that has been recently accepted, we decided to put a mix between gender, between farmer role in the seed system, and also getting some kind of information on their income and socioeconomic characteristics to create these 4 segments of farmers in these 3 African countries here. So was done with Amaranth. It was done in Benin, Mali, and Tanzania. And then we tested 14 World Veg GeneBank genotypes. And I think it was one local variety in each of these countries. But all of them GeneBank accessions, let's say the best GeneBank accessions after characterization in the GeneBank of World Veg. So then they decided to send it out to the farmers to see what the farmers think about these accessions. And then the farmers did the assessment, and then using socioeconomic data, we managed to create these 4 groups. So basically we have a group of what we call older women generalist. So basically women above 43 years. Most of this group is dominated by women. The average income from Amaranth is 22%. They have a very small, they don't have so many years growing Amaranth. They are older, but they don't grow Amaranth so often. And then basically they control both of the sale and production. And then here you have women specialists, and also older men generalists and young men specialists. And each of them have different characteristics. And this is the characteristics that influence the choice in the seed system. Here, for example, you in the paper soon you will be able to access this. But here you have the 4 groups by the preference. And here you have all the traits that the farmers assessed. So you have plant survival, marketability, and the first harvest, overall performance. And then you have here the varieties, in the let's say each of these is a PCA, a principal component analysis of the probability of one variety being the best among the others. And here you have, for example, that each of these arrows will point to one thing. So, for example, here we see that these men in this group, they are generalists for one thing. So basically, they don't have a clear preference towards a specific variety. For them, it doesn't really matter which variety they are growing. They have Amaranth as basically is not their main income, so it's only 14% of their income, and they are not really so much experienced in growing this, so they just want to grow because someone might have tell them that this is a cash crop and then they can get some extra money. So for them, doesn't really matter which variety they are growing. So here is an insight for the seed system. You can offer the variety that is very likely to be the higher yielding variety or high tasting variety for the next climate condition. So if you can drive your decision based on the climate resistant of this variety, not in other things, because this group doesn't really care about that. But here you have an other group, for example, which is the specialist, where you see the women specialists, they have a clear preference towards these five varieties. If you see, all the arrows are pointing to these five varieties here. So for some specific reason, they prefer this variety. So this is something to inform as well the system, the stakeholders, on which variety should be prioritized to be informed or to be available for them. So this is this is the fifth case now where we moved from an approach which is very, let's say, binary and dichotomous approach to something which is more creating specific groups or characteristics which can be easily informed or farmers can identify easily by seeing this. No? So one thing that we are moving now is basically creating personas out of this. So you create a persona like thing, and then the farmer will we can inform you, say, look, this is the characteristic of this persona. Do you identify yourself with some of these characteristics? And then we can start to provide the recommendations from that. So this is how we move now from gene banks to farmers, and also again to farms, when the farmers getting insights of what worked well and what didn't work well in the overall project. Most people, they say sometimes that the farmers may not get the best variety for them, and this is true. This is one of the limitations of Tricot, practically in the implementation part, but you need to look at Tricot not on the implementation part, but you need to look at from the, let's say, from the next 4 years when you start to provide recommendations over different cycles, because you can see from the big picture. Now you can take the big picture and see what worked well and what didn't work well for specific groups of farmers, and then start to provide recommendation. And then I come back to the first thing that I said, no, about data capturing. So sometimes the trials are designed to fulfill a specific goal of a trial or a project, but the idea here is also to fulfill this goal, but also to have the idea that you can do more with the same thing, the same amount of money. You can do more, and then you can get more insights after many interactions with the stakeholders. To summarize that, no, Tricot is a citizen science approach built on a multi-institutional partnership. So this is not something that is done exclusively by the Alliance of Bioversity and CIAT, but it's something that has more than 40 stakeholders involved. Of course, it enables scaling of decentralized trials by offering a robust experimental designing, straightforward data capturing approach. Farmers are exposed to a crop diversity and assess it under the local conditions. And on-farm and consumer preference data can be linked to several other data sets to explain how and why farmers consume and consumers take decisions. Here you can have links now to assess. This is the website of ClimMob here, here, climmob.net. And then you can access all these tools here. You know, thousandfarms.net is the where we have all the resources for the tricot, and you can get more insights by reading all the publications that I also put here in the slides. And of course, if you want to contact me, you can contact. This is my email, and I'm really happy that to present this presentation here, and also to share the experience. Again, this is not only me, I'm the one here talking, but there are many people involved in that, and I hope that they feel that they are part of this presentation as well, because this work is not possible without the support of the stakeholders. In that case here, we also had a lot of work with the World Veg and Wageningen University, and also the support of Crop Trust, the government of Norway, and Gates Foundation. And thank you all for the opportunity.
Thank you very much, Kauwe, for a very informative presentation and for showing us step by step how that tricot approach facilitates the evaluation of gene bank accessions through farmer-led decentralized testing. I think this approach is especially valuable because it brings gene banks and farmers together, which has been an issue in the past, to perform germplasm evaluations in a relatively quick, cost-effective, and I believe a scalable way. So thank you very much again for the presentation and for the examples. I think it brings everything together to show us specifically how you can analyze the data in different ways, as you have shown us. So that was very informative. To our viewers, thank you very much for watching this Grow webinar. We hope to see you in the next one. Goodbye and thank you. Bye-bye. Bye.